Imagine trying to light, a cozy, crackling campfire in your backyard.
That's nice.
Right. You've got your marshmallows ready to go, but when you reach for a lighter, you realize the only tool you have is a military-grade flamethrower.
Well, you get your fire started, technically.
Right, technically.
Yeah.
But you are also going to incinerate those marshmallows, melt your lawn chairs, and probably burn down the entire neighborhood in the process.
Yeah, that is a slight problem.
Just a slight one. But for decades, this has been the exact terrifying dilemma doctors face when treating severe human pain.
Exact.
The most powerful medical tools we have, the ones that are absolutely necessary for someone recovering from major trauma or invasive surgery or battling cancer, they also come with these catastrophic, sometimes deadly side effects.
So relief itself inevitably becomes the danger.
Exactly. So welcome to the Deep Dive Lab. Today, we are looking at a stack of recent research to answer a strange, almost impossible question.
Which is, why is it that the drugs that are best at stopping human pain are also the most dangerous?
Right. And how did scientists recently find a mind-bending answer by essentially picking up that exact molecular flamethrower?
Yeah, to understand how researchers finally approach this paradox, we really have to look at the underlying biology of how we treat pain.
Okay, lay it on us.
So conventional opioids, medications like morphine or fentanyl, they are the undisputed gold standard for pain management.
Because nothing else really works quite like them, right?
Right. Nothing else in our modern medical arsenal comes close to their raw efficacy. I mean, when someone takes an opioid, it travels through the bloodstream, crosses into the brain, and it binds to something called the µ-opioid receptor.
And that's often just referred to as the MOR, correct?
Exactly, the MOR.
So the drug acts like a physical key fitting into a very specific lock inside the brain. And once the key turns, the pain signals just stop.
That is the intended outcome, yes.
Right.
But turning that lock initiates a whole cascade of events inside the cell. It's not just a single isolated reaction.
Right, it triggers a chain reaction.
Exactly. So activating the µ-opioid receptor triggers a primary signaling pathway known as the G pathway. And this G pathway is the desired mechanism.
That's the pain relief.
Yeah. It effectively blocks the transmission of pain signals through the nervous system, which provides that profound relief.
It's essentially like flipping a light switch in your house to turn on the living room lights. The lights come on, the pain goes away.
That's a good way to look at it.
The problem is that the wiring in this house is completely crossed, because flipping that exact same switch to turn on the lights simultaneously triggers the fire alarm. It turns on the overhead sprinklers and automatically calls the fire department.
Right, and in the human body, that secondary internal fire alarm is a separate signaling route. It involves a group of proteins called beta-arrestin.
It's arrestins, okay.
Yeah, so when an opioid binds to the receptor, it doesn't just activate the G pain relief pathway, it also heavily activates that beta-arrestin pathway.
And that's where things go wrong.
Exactly. This secondary route is responsible for all those devastating physiological reactions we associate with opioids. Most notably, it causes respiratory depression.
Which is the really scary part.
Very scary. The signaling actively interferes with the brainstem's ability to monitor carbon dioxide levels. So it causes a person's breathing to slow down dangerously or just stop entirely.
And that is the primary mechanism of death in an overdose.
Unfortunately, yes.
And the beta-arrestin pathway also sabotages the long-term effectiveness of the drug too, right? Because these proteins physically grab onto the opioid receptors and basically pull them inside the cell.
Right, they hide them from the surface. And that process is the biological basis of tolerance.
Because if there are fewer receptors available on the surface of the cells, you need more of the drug.
Exactly. A patient requires increasingly larger doses of the opioid to achieve the exact same level of pain relief.
So you are forced to flood the system with more of the drug, which then triggers an even stronger beta-arrestin response.
Yes, which further suppresses the respiratory system.
And while all of this is happening, the drug is also deeply penetrating the brain's reward centers.
Right, triggering intense, rapid surges of dopamine that rewire the circuitry and drive compulsive cravings.
It really is a biological trap. I mean, you take the medication for severe pain, it slows your breathing, rewires your brain for addiction, and then literally forces you to take higher, deadlier doses just to keep the agony at bay.
It is a trap. And for a very long time, the prevailing scientific dogma was that this trap was an unbreakable package deal.
Like you just couldn't have one without the other.
Exactly. The medical community basically operated on the assumption that high-efficacy pain relief inherently required accepting high-risk, potentially lethal side effects. They were viewed as biologically inseparable.
But to find a way to separate them, a team of researchers decided to look at a class of synthetic opioids that were originally synthesized way back in the
Right,
1950s.
the nitazines.
And wait, nitazines? I mean, anyone paying attention to modern public health news will immediately recognize that name. Nitazines are those incredibly dangerous, highly potent synthetic street drugs, right?
Yes, they are.
The ones currently being mixed into the illicit drug supply, driving massive spikes in overdose deaths globally. I mean, some of these
Right.
compounds are estimated to be, what, up to 1,000 times stronger than morphine?
Yeah, 1,000 times. The history of nitazines is entirely counterintuitive to the idea of building a safer drug.
I would say so. Why on earth would scientists use those?
Well, during the 1950s, pharmaceutical researchers were actively synthesizing these compounds as potential painkillers. But they were quickly abandoned and permanently shelved by the industry, precisely because their potency was just uncontrollable.
So they were just too strong to be safe.
Way too strong. The risk of extreme, immediate respiratory depression was astronomically high. The therapeutic window, which is the difference between a dose that provides pain relief and a dose that causes lethal respiratory failure, it was virtually non-existent.
Okay, so scientists who are looking for a safer alternative deliberately chose a chemical class that was historically discarded for being too good at being dangerous.
Exactly.
Yeah.
They went straight for the molecular flamethrower.
That is wild.
They looked at that raw, unbelievable potency, that thousand-fold strength over morphine, and asked a very daring question. What if that extraordinary power could be harnessed and fundamentally tamed?
So they wanted to keep the pain-killing power, but completely engineer away the danger.
That was the goal.
And the actual execution of that idea is detailed in a landmark study published in the journal Nature.
Right. Dr. Juan L. Gomez and an extensive team of researchers from the National Institute on Drug Abuse, Stanford University, and several other institutions performed a highly targeted piece of molecular engineering.
What exactly did they do?
They took a wildly potent nitazine called etonidazine, and they made a single specific chemical modification. They attached a fluorine atom to the existing molecular structure.
Okay, just one atom.
Just one. And this modification created a novel compound called FNZ.
And then what happens?
Well, when FNZ is introduced into the body and processed by the liver, it rapidly metabolizes into a new, highly specific molecule. And that metabolite is called DFNZ.
And DFNZ is the compound that essentially breaks the biological dogma.
It is. The resulting data on DFNZ was so significant that it was actually the subject of a dedicated Nature News and Views commentary by researchers Victor Mathis and Emmanuel Dark.
So what did they find when they tested it?
When the research team tested this DFNZ molecule in rodent models, they observed that it acts as a, quote, superagonist.
The superagonist.
Yeah, meaning it binds to and activates the µ-opioid receptor far more strongly than even a full dose of conventional morphine. It delivers just an overwhelming signal to block pain.
Okay, but this is the crazy part. Despite being hit with a painkiller significantly stronger than morphine, the rodents didn't stop breathing.
They didn't. Their respiratory rates remained totally stable.
Wow.
And over time, they didn't build up a tolerance requiring higher doses either.
And when the research team abruptly stopped giving them the drug, the animals displayed almost no discernible signs of withdrawal.
It was incredible. A drug with supramaximal efficacy actually proved to be vastly safer than the conventional, weaker alternatives.
How is that even possible? I mean, how does a drug stronger than morphine end up being safer?
Well, the researchers discovered that DFNZ achieves this by utilizing three distinct, highly sophisticated biological mechanisms. It basically bypasses the traditional opioid trap.
Okay, let's break these down. What's the first mechanism?
The first one directly addresses the crossed wiring of the µ-opioid receptor that we talked about earlier. It's a phenomenon called pathway bias.
Pathway bias, meaning it favors one path over the other.
Exactly. Instead of the drug acting like a standard key that simply turns the lock and activates everything inside the cell, the unique physical shape of the DFNZ molecule forces the receptor into a very specific physical conformation. It twists the lock in a way that heavily favors activating the G signaling pathway, the pain relief route, while fundamentally failing to engage the beta-arrestin pathway.
Oh, I see. So it literally manages to turn on the lights without ever triggering the fire alarm.
Exactly.
Eh.
By maintaining the strong bias toward the G pathway, DFNZ delivers profound analgesia without initiating the cascade of secondary proteins that pull the receptors inside the cell and cause tolerance.
That is brilliant. But simply fixing the receptor's internal signaling doesn't solve the broader physical problem, right?
What do you mean?
Well, conventional opioids like fentanyl are notoriously dangerous because they easily cross the blood-brain barrier and physically pool in the brain tissue.
Ah,
They
yes.
just linger there, continuously suppressing the respiratory centers. So if DFNZ is a superagonist, wouldn't a buildup of that molecule in the brain tissue still be catastrophic, regardless of the pathway bias?
That is a great question.
Yeah.
But the blood-brain barrier is not just a passive physical wall. It's an active, heavily guarded fortress.
It has defenses.
Exactly. The barrier relies on efflux transporters, which are specifically a set of specialized proteins. Their primary biological function is to patrol the barrier, identify foreign or potentially toxic substances that have slipped into the brain environment, and physically pump them back out into the bloodstream.
Oh, wow. So think of these transport proteins as highly aggressive bouncers at an exclusive VIP club.
That is exactly what they are.
DFNZ manages to slip past the velvet rope and enter the brain just long enough to deliver its massive pain relief signal to the receptors.
Right?
But the moment that signal is delivered, those specific transport proteins, the bouncers recognize, DFNZ grab it and forcefully eject it back into the bloodstream.
Yes, before it has any time to accumulate and shut down the respiratory centers.
Do conventional opioids not do this?
No, conventional opioids do not trigger this active ejection. Fentanyl essentially evades these specific bouncers. It enters the brain and remains there, which is why it causes such severe, prolonged drops in localized brain oxygen levels.
That makes so much sense.
But DFNZ is an active substrate for these efflux transporters. The research team proved this by measuring the distribution of the drug. The concentration of DFNZ in the blood plasma was vastly higher than the concentration inside the brain.
So it gets in, triggers the superagonist pain relief, and is immediately expelled.
Exactly.
But wait, if the transport proteins are actively kicking DFNZ out of the brain so quickly, that has to fundamentally alter how the drug interacts with the brain's reward centers too, right? Which is the core driver of addiction.
You're spot on.
Uh-huh.
And this leads to the third critical mechanism, which centers on the distinction between tonic and phasic dopamine release.
Tonic and phasic.
Yeah. Addiction is basically a process of extreme neurological training. When a person consumes an addictive substance like heroin or fentanyl, the drug deeply penetrates a specific reward center in the brain.
Okay.
And this triggers an explosive, massive, and rapid surge of dopamine.
A phasic release.
Yes. A sudden floodgate opening that overwhelms the brain's circuitry. And that massive, instantaneous rush is what chemically teaches the brain to associate the substance with an overwhelming reward.
Creating those deep neurological grooves that drive powerful compulsive cravings.
Exactly. But DFNZ completely alters that release profile.
How so?
Using advanced biosensors implanted in the brains of the rodent models, the researchers observed that DFNZ does not trigger a phasic flood. Instead, it only causes a slow, continuous, steady trickle of dopamine.
And that's the tonic dopamine release.
You got it.
And the mechanism behind that trickle is fascinating because inside that specific reward center, the µ-opioid receptor doesn't operate in isolation.
Right. It frequently pairs up with an entirely different protein called the gallinin-1 receptor. They physically link together to form a complex.
So when a conventional opioid hits that paired complex, it initiates the massive downstream reward signal.
Yes. However, DFNZ interacts very weakly with this specific µ-opioid gallinin heteromer. It basically fumbles the molecular handshake.
I love that phrase. It fumbles the handshake.
Yeah. Because of that weak interaction, the complex essentially acts as a highly restrictive valve, dampening the downstream signal and preventing the massive dopamine hit that creates the addiction loop.
And the researchers actually put this to the test, right? With self-administration trials, where the rodents were given the ability to press a lever to receive the drug.
They did. And the animals pressed the lever significantly less for DFNZ than they did for heroin.
And crucially, when the drug was completely removed, their drug-seeking behavior extinguished very rapidly. They did not relapse.
The inherent addiction potential of the molecule was drastically lower.
Which is just phenomenal. And you look at the synergy of these three mechanisms, the implications for pharmacology are just monumental.
They really are. The current global opioid crisis is a devastating public health emergency, and it's fueled entirely by medications that invariably hijack the brain's reward centers and relentlessly suppress the respiratory system.
Right. Hundreds of thousands of lives have been lost because the medical community believed that high-efficacy opioids simply could not be safe.
And this discovery completely shatters that foundational assumption.
It's amazing. Scientists took a class of drugs deemed too volatile for human use, added a single fluorine atom, and engineered a molecule that features built-in safety brakes.
Built-in brakes, exactly. It biases its signaling away from the dangerous beta-arrestin pathway. It actively utilizes the brain's own transport proteins to prevent toxic accumulation, and it restricts dopamine release to a slow trickle.
All to avoid hijacking the reward system.
Yes. It proves that supermaximal efficacy pain relief that vastly exceeds the capabilities of morphine can be achieved without triggering severe adverse effects.
Think about what this means for you or, anyone facing a severe medical intervention.
It's life-changing.
A patient recovering from invasive surgery or enduring the agonizing pain of late-stage cancer currently has to navigate their recovery with this terrifying shadow looming over their hospital bed.
Yeah, there is the constant ambient fear of an accidental respiratory overdose, or the deep anxiety of developing a lifelong opioid use disorder simply because they required medical relief from profound physical agony.
If a molecule operating on the principles of DFNZ successfully reaches the market, it could entirely eliminate that shadow.
The medical intervention would just be pure, uncompromised relief.
Which is an incredibly hopeful vision for the future of pain management.
It is. However, we must introduce a necessary and stringent reality check here regarding the timeline and the process.
Definitely.
The medical community cannot afford to repeat the catastrophic tragedies of recent history. The authors of the study, as well as commentators Mathis and Dark, explicitly warn against premature optimism.
Right, because the modern opioid crisis was accelerated in the 1990s by the aggressive promotion of drugs like oxycodone,
Exactly.
which were marketed as safer, less addictive alternatives to traditional opiates. But those early, confident claims obviously lacked the foundation of rigorous, independent, long-term evaluation.
Yeah. The data surrounding DFNZ is undeniably promising, but it is currently promising in highly controlled rodent models.
Just rodents for now.
Yes. The researchers themselves emphasize that this study represents a foundational first step, not a finished product ready for human pharmacies.
Because a molecule like DFNZ requires years of exhaustive, multifaceted testing.
Absolutely. For instance, the current research did not fully explore sex differences in how the novel compound is metabolized, or how it might differentially affect behavior across demographics.
Right. Furthermore, they really need to evaluate its addiction potential and efficacy specifically within chronic pain models.
That's a huge one.
Because the central nervous system that has been rewired by chronic long-term pain reacts to chemical stimuli very differently than a healthy acute pain nervous system.
Exactly. And while DFNZ proved vastly safer than fentanyl, it's not completely devoid of side effects at extreme limits.
Oh, really?
Yeah, when administered at doses far beyond what was required for maximum pain relief, the drug did induce catalepsy, which is a state of severe muscle rigidity.
Oh.
And it also caused a measurable drop in core body temperature. So it is a brilliant feat of molecular engineering, but it is a potent chemical that requires deep respect and a lot further mechanistic investigation.
Still, the journey of this research fundamentally changes the paradigm of what is biologically possible.
It completely flips the script.
I mean, we started with a molecular flamethrower, the nitazines, a drug class from the 1950s that was permanently blacklisted for its tendency to cause immediate respiratory failure.
But instead of abandoning that raw power, scientists studied it at a microscopic spatiotemporal level.
And by making a tiny structural tweak, they discovered DFNZ, a compound that uses the brain's own active defenses and clever cellular signaling to basically turn a reckless flamethrower into a highly targeted, infinitely
solution to a seemingly insurmountable public health problem isn't always to retreat from a dangerous path.
Sometimes you just have to keep going.
Right. Sometimes the solution is to walk directly down that path, understand the exact signaling mechanisms of the cells involved, and literally rewrite the rules of pharmacology to tame the danger.
Which leaves you with a lingering, thrilling question to mull over. If researchers can take a notoriously lethal molecular flamethrower and engineer it into a uniquely safe, super-powered painkiller.
Think about the possibilities.
Exactly. What other discarded, dangerous, or failed molecules are sitting on laboratory shelves right now?
There could be hundreds.
Right. What other toxic compounds from decades past are just one tiny chemical tweak away from becoming the medical miracles of tomorrow?
It's an exciting thought.
It really is. If this discovery is just the beginning, what might scientists uncover next? Thank you so much for joining us in the Deep Dive Lab. If you love unraveling these scientific mysteries with us, be sure to follow and subscribe and keep your curiosity alive. We will catch you on the next deep dive.